Hello and welcome to this week's What Were They Thinking? We begin with something of a nuclear bomb, or at least the results of testing of nuclear bombs in the 1950s. The nuclear weapons testing of that period was widespread and conducted by many countries, but in particular America, who were trying to see whether or not their weapons were effective and what kind of yield they would get. This resulted in a lot of radiation being released. The radiation affected the salt water in the ocean. This resulted in the chlorine becoming ionized and in turn altering the isotope. This created chlorine-36, a radioactive isotope that was released into the air and eventually became caught up in the ice sheets of Antarctica. In the end, we are now seeing the fallout from this more than 50 years later as these same ice sheets begin to melt. Researchers use chlorine-36 as a means of dating Antarctic ice sheets and other locations. This is because it becomes trapped in the snow and ice under the surface. Gradually, it gets buried further and further. This is why the chlorine-36 levels have been reduced to the point where they were before this worldwide testing of nuclear weapons. The problem researchers have found is that those areas that have high and low snowfall each year have varying levels of chlorine-36, and some of them have high levels very near the surface, which could, as said earlier, result in its release to the atmosphere. Researchers at the Russian Vostok research station have found that the areas around their research base have levels of chlorine-36 10 times the background level. In another nuclear bomb of sorts, scientists have discovered gender-specific brain cells in the brains of mice. It is generally accepted in biology that most species are sexually dimorphic. This means there are two sexes that generally express anatomical, physiological, and biological variation with some degree of overlap. In the case of humans, that has much to do with the reproductive organs and secondary sex characteristics. In mice, similar changes occur, but, at least in humans, there has been a great deal of contention over how much difference there is between the sexes and whether or not it is simply a binary. The researchers from the California Institute of Technology were looking at the brains of mice, and specifically the ventrolateral subdivision or the ventromedial hypothalamus of males and females. It is a very, very small structure of only 4,000 cells. Past researchers found that this structure has an important role in metabolism, sexual, and social behaviors. Having only 4,000 cells, the researchers were able to use a method called single-cell RNA reading technology, which tells them what genes are being actively used in those cells. This is distinct from more broadly used tissue samples of DNA, which will tell you the gross expression of what is going on in that tissue sample, which will often be made from mixed cell types. In this case, nearly all the cells are neurons, and they all relate to those three roles: metabolism, sexual, and social behaviors. These three roles fell into 17 distinct categories of cell activity in that part of the brain. That was found using another method called fluoro in situ hybridization, sometimes called the acronym of FISH. These two methods allowed the researchers to not only identify the distinct clusters of cells, but be able to figure out what was being expressed differently in each cluster. And, further to this, the way those cells split among the two sexes was distinct. The first, and already established, is that at least one enzyme was only present in male mouse brains. What was not known is that there was a specific cell cluster only present in the female mice. As mentioned earlier, there are physiological, anatomical, and sex characteristics that are gender-specific. That in and of itself means that some variation should not be a surprise. What is important here is that not only do you have cells expressing distinct, sexually defined differences, but that they are in large enough clusters that the whole tissue could be considered different between the sexes. These tissue differences were not directly derived from sex chromosomes. This further confounds and makes this an interesting finding, and, as mentioned earlier, something of a bombshell. In news related to the bombshell of sexually defined brains, we may understand why studies like that might not always translate into a human study. Animal research of any kind has a relatively low rate of conversion into a human model, even with a humanized animal model. This is because there is a fundamental underlying difference in the species that is not always easily overcome, even with the best of efforts. Brain studies are especially notorious, and one piece of research that has come out recently lays the blame squarely at the feet of the cerebellum. That is, part of the brain that receives and coordinates sensory information. The core of the problem the researchers found was that between conception and 9 months after birth, the cerebellum develops in distinctly different ways between humans and mice. Even our closest primate relatives demonstrate a distinct and different development through neoteny of the cerebellum. This leads to whole groups of cells that are present in the human brain but not in other species. The cells that are most relevant are normally only found in the cerebral cortex and are involved in memory, cognitive function, and language. Finding these in the cerebellum raises many questions. There is also another area specific to the cerebellum called the rhombic lip, which has progenitor cells that become the primary source of brain cells. These generally make up 80% of all neurons in the human brain, and development of this region is protracted, especially during gestation, where the cerebellar volume will increase 5-fold. This means that area of the brain must be producing more cells than ever, and the protracted period of time helps to accommodate this. Any alteration in that area can lead to significant developmental and neural defects that then lead on to neurodefects later in life. Diseases that are incredibly difficult to model. Comparing this with the development of a mouse brain, we can begin to see why some findings in an animal model with mice and the human condition do not match up effectively, as that portion of the brain completely diverges very early on in the development of life. Even if genes and other parts of the animal are highly conserved, you cannot model a disease in one animal if that animal simply does not have that anatomical, physiological, or biological function, as is the case of certain diseases that rely on that part of the brain. In other genetics-related news, the work by Dr. He in China with the CRISPR babies was heavily criticized when it first came out, with some researchers stating that they believed his work would likely lead to the premature death of the newborns. The researchers who produced that particular paper in Nature have now retracted their work due to what they describe as a data sample that left too much room for doubt. The original claim was based on analysis of data from the UK's Biobank. This is a huge genetics database where they were recording the prevalence of something called the CCR5 gene, which is a white blood cell receptor that makes them intrinsically vulnerable to HIV. The Biobank data set indicated that those with one copy of this modified gene were 21% more likely to die early. The CRISPR babies had both copies of this gene modified and were therefore at a higher risk. These claims were met with skepticism through peer review, and other researchers tried to reach the same conclusion using similar methods, the same databank set, and other nations' data. They could not replicate the findings. There are a number of reasons for this, and perhaps the most obvious is high throughput data collection of genetics is prone to producing some errors. This means some people will show up as positive and some will show up as negative when they should not. The researchers who made this original claim concluded that with such an error-prone data set, they could not make a definitive conclusion and as a result retracted their work. In another example of researchers revising how they and others view the world, some recent research coming out of a collaboration between America and Germany indicates that mitochondria might function differently than currently accepted. Like a standard battery, the mitochondria moves ions through a membrane. The transition of these ions is what allows energy to be made and stored for future needs. The shape of the mitochondria creates more internal folds, which then allows the greater transmission of these ions between the membranes, which in theory increases the efficiency of the mitochondrion without increasing its overall size. Very few people have had reason to question this accepted description of how the mitochondria functions. Generally, it was thought that cells have many mitochondria on the basis that if one were to fail, the others would pick up the slack, much like a standard battery that when it runs out can be swapped out for a new one. The researchers decided to see how true this was, since some cells have only a few mitochondria and the loss of a single one could be catastrophic. They were doing a number of things, using high-resolution microscopy and staining the cells to observe the fine details. They were then able to see how the voltage moved along and around each of the cells and to see if there was a universal charge or variation between cells and within a single cell. What they saw leads them to believe that mitochondria are more like a pack of batteries than a single battery. Think of it as being like a lithium charging pack. There are a number of lithium power packs within the outer plastic casing. Each of those carries its own charge, and that is what the researchers saw with the mitochondria. What they found is that each of the folds within the mitochondria acts independently from the others, meaning it can store and process energy without affecting the other folds of the mitochondria. This is the same way that a lithium charging pack can have one of the cells fail, but all the other cells within it will continue to function, but the overall unit will have lost some of its capacity but remain functional overall. In unrelated archaeological news, Egyptian officials have announced the excavation of 20 sarcophagi. What is important here is that they were both intact and sealed. Egypt has lost many of its archaeological artifacts over the last centuries due to environmental concerns, grave robbers, and developments. Movement of time in history has slowly destroyed some of Egypt's heritage. These 20 sarcophagi are, if nothing else, an amazing discovery that will provide much evidence and information about Egypt's own history. These artifacts were found in the site of the ancient city of Thebes. This was mostly occupied by the 11th dynasty, which ran between 2081 and 1939 BCE. This does not necessarily mean that they come from that period of time, and officials have yet to state from when they believe the coffins were made and buried. In more disconcerting news, Japan has imported Ebola, among other rather nasty diseases, in preparation for the 2020 tourist influx for the Olympics. Many of the diseases that have been imported to Japan for the first time in about 100 years all require the biosafety level 4 that was recently discussed in relation to the Russian facility that caught fire. Japanese officials state that they were imported in preparation for the possibility that tourists entering for the Olympics might bring with them these diseases, and Japan must prepare appropriate countermeasures in the event of an outbreak. Naturally, this has led some of the local inhabitants of the areas where these particular pathogens are to be kept to express concern, especially about potential outbreaks. Japanese authorities have replied by stating that they have not only the approval of the government, but that of the local authorities who they are working with to ensure that any benefits gained from this will significantly outweigh any possible risks. In a case of fantasy and fiction being closer to reality than we think, a number of legume crops have been grown successfully in a soil media similar to that found on the Moon and Mars. Being able to grow crops on the Moon or Mars one day is an essential element of being able to colonize these planets. This avoids the need for shipping massive quantities of food that would be a logistical nightmare, and quite possibly lead to some of the most unimaginative diets in the world, or is that in space. They tested 10 different species. All of them produced edible content except for the spinach. The findings from this study have been published in a peer-reviewed journal called Open Agriculture. The key difference between the Moon and Mars was that Mars produced results that were acceptable. The Moon produced poor results. The two different media are based on samples and analyses conducted both by various Mars landers and from samples taken from the Moon itself. While traditional soil cultivation is certainly the best option at present, primarily because it requires no extra equipment be bought, other options do exist, such as hydroponics and aeroponics, options that are being considered by various researchers in their own right. In similar Moon-based news, Elon Musk has admitted there is a potential major flaw in SpaceX's moon base plan. SpaceX planned to use the moon for several things. One was as a refueling point for their Starship, either on its way to other celestial destinations, or, more likely, on its way to Mars. The idea was to give the Starship just enough fuel to get from Earth to the moon. This is because leaving Earth is much harder than leaving the moon due to gravity. Being able to reduce the amount of weight the Starhopper needed for fuel meant it could either carry more materials or be cheaper to deploy from Earth. The question then became, how would they fuel these craft on the moon? And they had planned to be able to get carbon on the moon and convert it into fuel, particularly if they could find hydrogen and oxygen. This refueling station would then be the easiest way to deploy the same Starship on its next leg of the journey, being able to leave the lunar gravity field that much more easily. A mine on Earth dedicated to anything like this could take years if not decades to build, and SpaceX hopes to have something viable set up in the relative near future. While SpaceX is busy considering the possible barriers to deploying a moon base, they're also requesting approval to continue the deployment of their Starlink network with another 30,000 satellites. The Starlink network already has a number of satellites in orbit. The intention behind this network is to provide high bandwidth access to the world through satellite communication. This would remove some of the current barriers of physical cables in the ocean and underground. However, deploying this many satellites requires a number of things, not the least of which is approval from the FCC, and then filing this and getting approval with the International Telecommunication Union. A request to deploy 30,000 satellites in varying stages might make them question that approval. If SpaceX were to be successful, they would effectively have single-handedly cornered the satellite internet connection market. In a piece of old history and engineering genius coming through, one of Da Vinci's idle scribbles that is over 500 years old for a radical bridge has been shown to not only be viable but structurally sound. The bridge was designed in 1502 for Sultan Bayezid II, who was the head of the Ottoman Empire, and it was meant to connect Istanbul and Galata. Ultimately, the Ottoman Empire went another route, but not before Da Vinci made his design. Unlike bridges of the same time, it doesn't rely on massive arches, but is instead a single bridge going from shoreline to shoreline. If it were built at the time, it would have been the longest bridge. The interesting part of this was the researchers created a 1 to 500 scale model by creating 3D printed blocks and then assembling them to match the overall design. This used 126 pieces. You might think that simply assembling a model is not that impressive until you understand that what they built used no adhesive or glue. In fact, the original design required none of these, as it is all held together with compression. Finally, to add to the reasons why you may no longer have hope in humanity, in a case study coming out of urology case reports, a 22-year-old male presented at a clinic in Saudi Arabia without stating any symptoms. What he had was a pair of 8 centimeter long tweezers inside his urethra. and they had been sitting there for 4 years. He admitted to self-inserting them into his penis. The doctors note that this is not necessarily an uncommon practice, with psychiatric, intoxicated, confused, or sexually curious individuals doing so. What is unusual here is the item in question, with tweezers being found only once before in the medical literature. The patient reported no difficulties in urinating, which may explain why it took so long to present for some kind of resolution. Doctors identified both the location and orientation of the object by palpating the anatomical region and x-raying it. They then put the patient under general anesthetic. They were then able to find the tweezers quite some ways into the urethra, force them closed, and in a slight twist of irony, use a second pair of forceps to remove the first pair. Due to good fortune in the design of the tweezers removed, and the way it could be removed, the patient was able to avoid invasive surgical procedures. The man was able to return to urinating normally and leave in relatively short order. Given that it had been 4 years, the medical team was rather surprised at how well he could deal with the pain from having this object inserted as and how it was. They suggested that the patient undergo a psychiatric evaluation, which was declined, and no further follow-up was possible. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.